Hello, my name is Fatoumata Diallo. I'm a senior at Brown University, concentrating in health and human biology, and I had the wonderful opportunity to be a NIH SenNet CUSP intern at Dr. Königshoff's lab at the University of Pittsburgh Medical Center, working under the Pulmonary Sleep Critical Care Division. And today I will be talking about lung fibroblast senescence in chronic obstructive pulmonary disease. Cellular senescence is the irreversible cessation of cell growth and development. And however, the cell is still metabolically active. And this is triggered by environmental and genetic factors such as telomere dysfunction, activated oncogenes due to mutations and overactivation, and DNA replication stress, oxidative stress that can be induced by smoking, and cell-to-cell fusion. So short term, we know that senescent operates as a tumor suppressor and can limit tissue damage and is important for embryonic development. However, if these senescent cells start to accumulate, long term, they have this senescence associated secretory phenotype, well known as SAS, where it secretes a bunch of proteins like pro-inflammatory cytokines, chemokines, proteases, and growth factors that can lead to cancerous cells and age-associated diseases by accelerating the biological aging tissue process. And one of the age-associated diseases that is extensively studied in our lab is chronic obstructive pulmonary disease. It's a heterogeneous disease that consists of non-fully reversible airflow limitation, chronic bronchitis, and emphysema. Symptoms include shortness of breath, sputum production, cough, chest tightness, and more. And in our lab, we're mainly focused on the emphysema aspect, as you can see in this figure below, that is the abnormal enlargement and dilation of our tiny little air sacs, known as our lung alveoli, which is due to destruction and damage of our alveolar walls and connective tissue. And we're mainly focused on the molecular etiology of what causes the emphysema. And this paper establishes that cellular senescence is indeed involved in the pathogenesis of COPD. And the researchers' goal was to identify markers of aging and severely onset and older COPD-derived lung fibroblasts. I will note that the fibroblast cells are specialized cells that was my interest cell in my research focus this summer, which is a mesenchymal-derived cell that plays an important role in inflammation resolution and alveolar repair and regeneration by producing extracellular matrix contacts and collagen. And in this paper, all SCO-COPD patients were constituted as patients that suffered from COPD at the age of 52 and early, and they suffered from severe emphysema, and all COPD subjects were age and sex control matched. And as you can see to the right, the data shows, figure A shows a senescence associated beta galactase assay after 7 days post PQ treatment or untreatment. PQ is an environmental toxicant that can induce senescence. So as you can see here to the right, we have the SEO COPD and the older COPD cohort showing more blue precipitate, indicating more cellular senescence, as the beta galactase is a hydrolysis reaction that is only found in senescent cells at a pH of 6. And figure B shows the quantification of this. In total, with the control groups, you can see that there's less SA beta gal positive cells compared to the COPD. And this is just like separating them into their subgroups. And we also see a drop in cell count in the COPD cohort compared to the control, which makes sense because we don't expect to see proliferation once senescent cells are introduced. And then figures D and E show the SenNet's widely employed gene biomarkers of senescence P16 and P21, where it's increased in the COPD cohort compared to the control, all indicating that DNA damage and senescent-associated markers were more highly expressed in the COPD-derived fibroblast compared to the control subjects. So one question that still remains is what are the underlying mechanisms that cause these fibroblast cells to become senescent? One theory is that the Wnt signaling and WIF1 dysfunction and dysregulation can cause these fibroblast cells to become senescent. The reason being is that the Wnt signaling pathway tightly regulates the cell-to-cell crosstalk between AT2 cells and fibroblasts that I will expand upon in the next slide. However, here, the way that the Wnt signaling pathway works is, as you can see from the off state and the on state, we have Wnt ligands available to bind to the frizzled complex and the LRP56 receptors, inducing a signaling cascade that eventually phosphorylizes the destructive complex here, which inactivates it, allowing beta-catenin to accumulate in the cell, then eventually translocate into the nucleus, where gene transcription will be turned on. And the way that WIF1 works is that it's an antagonist that binds to the Wnt ligands, inducing a conformational change that doesn't allow it to bind to the frizzled and LRP56 complex. So the destruction complex will degrade the beta-catenin, so transcription will not be turned on. And as we can see here, axin 2 is a downstream target gene that is reduced Wnt signaling, so we can use that as a biomarker to assess Wnt signaling in the cell. The reason why this is important is because it's well established that Wnt signaling is severely downregulated in COPD, and using RNA single cell sequencing dataset published early this year in our lab, we were able to identify that AT2 cells AT2 cells secreted WIF1, and they were overexpressed. And in other disease contexts, it was shown to induce cellular senescence, and we would like to see if that holds true in our COPD context. So my goal of the summer was to decipher paracrine effects of WIF1 secreting AT2 cells in the fibroblast microenvironment. So as we can see here in this diagram, we have the alveolar type 2 cells that are epithelial derived. They're cuboidal shaped, and if there's normal beta-catenin signaling, they have this progenitor capability to differentiate into an AT1 cell post acute injury to help regenerate the alveolar, or they can proliferate and renew as another AT2 cell. So here we're interested to see at a certain cell state, we know that AT2 cells secrete WIF1, and we want to know if it disrupts this cell-to-cell crosstalk in a manner that induces senescence to the fibroblast cells. Another component of our experiment was looking at the canonical Wnt3A ligand and seeing if that has a direct effect to the fibroblast cells as well, which can, in a manner that induces senescence, because we will have a conditioned media where we have the Wnt3A ligands available with WIF1 and without WIF1. So my hypotheses include fibroblasts exposed to WIF1 will express increased levels of senescent biomarkers such as P16 and P21, and there will be significantly reduced expression of Wnt signaling exhibited in the COPD fibroblast cells. So the techniques I learned this summer was cell culture, which consisted of seeding our COPD and healthy primary fibroblast cells. The primary fibroblast cells indicate that they were isolated from a previously living organ and tissue. And one key factor of cell culture is sterility, which helps us minimize unreliable experimental results. And by working under the tissue culture hood when dealing with our cells, another technique I learned was qPCR, which is allowing us to amplify DNA to see the relative gene expression changes in our P16, P21, and axin 2 gene. And our treatment groups consist of our base media, which is just like a media of growth factors that help the cell grow in culture, and the base media plus WIF1, our inhibitory factor, and then our conditioned media that contains the Wnt3A ligand I mentioned earlier, and the conditioned media plus the WIF1 protein, and then bleomycin, another environmental toxicant that can induce cellular senescence. Lastly, I learned the cyclone assay, which helps us quantify cell survival and division. Our workflow includes that day one, we serum starved our cells to synchronize them to all the same cell state, and then treated them with every 24 hours using 100 nanograms per milliliter of WIF1 to a 1:5 ratio of Wnt3A. After the first 6 hours, we would collect our cells, our healthy cells and COPD cells. Here we have an N of 1 with 2 technical replicates. And then after 72 hours, we would repeat the same process, collecting our cells for RNA extraction for the qPCR or the cyclone assay to read our cell viability. So the experimental results for the cyclone assay, we normalized everything to our untreated group to see the relative changes. And it was interestingly see that in the healthy, after 72 hours, we noticed that there was roughly a 20 to 30% drop in our condition media plus WIF1 group. And that was just exacerbated in the COPD treatment group, which is interesting to see how WIF1 does and indeed affect the proliferation of the fibroblast cells. For our qPCR results, as we can see at the top, which measures the AXIN2 gene, we have the 6-hour AXIN2 healthy versus COPD, where we can see our positive control, which is the Wnt condition media group, because we know through previous studies and research, we expect to see an increase in AXIN2 because Wnt signaling should be upregulated. And we see that it is working here in the COPD 6-hour. And then once we get to the 72-hour AXIN2 COPD, we see an increase in the Wnt condition media again. However, the magnitude of change for the Wnt condition plus WIF1 group is less exacerbated compared to the 6-hour. And then on the bottom panel, we see that there is, for the P221 gene across all groups, which is a direct measure for senescence, the bleomycin positive control was working because it was severely upregulated. This was not seen in the P16 gene. That's why I didn't decide to include it in our final analysis. And I would like to note that in the P21 COPD 72-hour, it kind of aligns with our hypothesis that we would expect to see more senescent cells when we introduce the WIF1. So as we can see, we don't know much of what the base media effect has with WIF1, so it's relatively low together. And then the Wnt3A condition media group also seen more senescence compared to the base media treatment groups. And then we saw an increase as well when we added the WIF1 protein, and then the bleomycin, it obviously shot up. So for our conclusion and future directions, short term, we should increase the number of COPD in healthy subjects and time points to build more conclusive data. And then long term, we would run a similar experiment using Wnt5A, a non-canonical Wnt ligand that is known to be expressed in fibroblasts as the condition media to see how the data may differ. With that, I'd like to thank the NIH SenNet CUSP internship program for accepting me into this invaluable research opportunity. Dr. Cornfish, my PI, and Cassandra, Brian, Pooja, and Haley, my COPD squad that taught me a lot this summer and helped me grow as a scientist, and the Pink and Blue Lab for being so welcoming and supportive of me as well.